what were the two hormones we were talking about which are being released from the stomach, cells in the stomach? Gastrin and great, good, good, good. What is the receptor via which gastrin acts? Good, 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 good. And CCK stands for what? Cholecystokinin. And then what are the two hormones which we come across when we talk about duodenum? Secretin and CCK. CCK. Secretin and what? CCK. What is the chemistry of all these hormones basically? They are all peptide hormones. They are all peptide hormones. Good, good, good. And the secretin and CCK together they bring about the release of pancreatic juice from the exocrine pancreas cells and the bile from the liver, gallbladder actually, and of course the bicarbonates, etc., etc. Okay. Stop now. We'll move on. I'll show this to you at the end also, but let's now move on with our lecture. So what we are doing right now is we are trying to familiarize ourselves with this very interesting organ called, what do you call it as? What do you call it as? Liver. What do you call it as? Liver. We have heard of it, liver. Now what we are going to do in another 5 minutes is try to understand the anatomy of liver. It's very basic, yeah. Oh, excuse me, I'll do it now. So in the previous lecture we said that it's made up of what? Lobules. Lobules. What do you call them as? Lobules. They're cylindrical, not exactly, with 6 faces, and then it tapers. And the one lobule, another lobule, another lobule, another lobule, thousands of them, they make the entire liver. Okay. And then the author gives it here as this anatomy of a single lobule. Okay. And then we have seen that the liver has blood supply from 2 sources. What were they respectively? One is hepatic portal and one is hepatic artery. Hepatic artery is mainly for oxygen, okay, and hepatic portal is to bring a lot of food material other than fats from the intestine to the liver. Okay, so far. And we have seen that at every angle or at every corner of the lobule, we see 3 tubes. Okay, and what were the 3 tubes? The 2 of them are blood vessels. One of them is a branch of this is a branch, it's blue, it is hepatic portal, it is, and then there is another this branch of hepatic artery, and the third green one is for what? Is for bile. Okay. Now what I am going to tell you is very interesting. Follow the story. So I said that each lobule, okay, each lobule will consider as a cycle wheel, and through the center of the cycle wheel, okay, now it's actually not like a wheel. If you take a transverse section, it's a wheel, okay, but if you see in 3D, each wheel is like a wall. So, one wall, second wall, third wall, fourth wall, and all the walls are projecting towards the center of the wheel like the spokes of a bicycle wheel. Are you with me? And at the center, you have a very major vein. At the center is the vein. That is called as central vein, that is also called as hepatic vein, and that will carry the blood away from the liver to the vena cava and to the heart. So, how is the blood going from the liver back to the heart? It is vena cava. Are you okay so far? All right. Now we have, okay, now I am going to talk about the wall, vertical wall. So, in the lobule, vertical wall. Now, each wall is made up of bricks. Each brick is a cell. Okay, so far, each brick is a cell. Okay, now this is a wall around me, all around me. So, one cell, second cell, third cell, fourth walls, you make a huge wall. Okay, wall everywhere. On this face, on my right, there is a tube in which I pour bile, and on my left, there is a capillary. Okay, so far. Okay, where is on this side? There is a corner of the lobule. The central vein is somewhere there. So here in my corner, there are two tubes. One of them is bringing the hepatic artery, branch of hepatic artery, that is bringing oxygenated blood, and next to that is a hepatic portal, and both of them are pouring blood into the capillaries and tiny capillaries, and those capillaries are flowing this way. Where are they going? They are going there in the center somewhere of the lobule. From there they will get into the central vein, hepatic vein, and that hepatic vein will drain to heart. So in this, so whether this is a red one is the branch of hepatic artery or the blue one is a part of hepatic portal vein, the blood is flowing this way, this way, this way, and then this way, and then this way, and then back to the heart. Hundreds of lobules, hundreds of them. Is it very clear? Okay. So from the, so let us see if you are somewhere, where are you? You are in an RBC. Where is that RBC? In the blood vessel. Where? In the intestine. Okay. So you run along. Where do you run along? You run along, you enter, you find yourself in hepatic portal system. Then you run along, finally you will find one of here. And then you get into the liver, okay, and then you travel, okay, on right and left there are walls, there are cells, hepatic cells. You go through a capillary, go to the central vein, okay. You suddenly find that you are joined by another, capillary. That capillary is bringing the arterial blood from the hepatic artery. Let me try to clarify this. So this is the red vessel, it is bringing what kind of blood? And here is a blue vessel, what kind of blood? The capillaries, both of them, as they go towards the central vein, they mix with one another. The blood mixes. So the blood coming from two sources, okay, those capillaries coalesce with one another, okay, and then the mixed blood will pour into the central vein and then back to the heart. Is that very clear now so far? Now, so this is a wall. On the left are the capillaries. In the capillaries, I am either getting a branch of the hepatic portal vein, or I may be getting a branch from the hepatic artery, or I may be getting the mixed blood. Fine, I am fine with that. On the right, I am a hepatic cell, I secrete bile, and that bile goes into the bile duct. Now listen to this. That bile, the bile flows in the opposite direction. In what direction does it flow? Opposite, means those cells. Now let us see here. Okay. So okay, I will draw your attention now. Can you see these cells? The green is indicating all this green is indicating the flow of the bile. And these cells, I think I can show this to you better in my next slide maybe. Hope I can do a better job here. I think I can do a better job here. Okay, good. Now can you see a wall like this that goes to almost to the center? So where am I? I am at one of the corners. Okay, and there is a wall which is actually plate of cells. They go all the way towards the central vein, but they do not open there, they are closed here. So they form a V. Are you with me? And on the inner side of the V, they will secrete bile which flows in this direction. It gets into the bile duct, okay, and then it goes to the gallbladder, okay, and then the common bile duct, and then it opens into the duodenum. Beautiful anatomy. Look at the cell. On one side the blood flows in one direction, on another side the bile flows in the other direction. Is it very clearly absorbed? Now this anatomy, okay, anatomy is amazing. The whole functioning of the liver, okay, is based on this amazing anatomy of this organ. Now what happens is, that is the most efficient way for the cells to keep on getting the material on one hand and then process it and then store whatever you can, whatever enzyme process. At the same time you keep on synthesizing bile. Okay, so just making it make sense, and then therefore for every cell you have to have access to a bile duct, and that tiny bile duct which comes from a cell joins the bigger bile duct. That bigger bile duct will flow into the bile duct which is at the corner of the lobule. So for you try any other way and find that this is the most efficient architecture of the liver. Okay. Now the blood that comes from the hepatic portal system is the blood that is coming to hepatic portal system. That blood has already been through the capillaries in the intestine. So the blood flow is very low. The pressure is very low. The pressure is actually found to be 8 or 9 mg. The blood that comes from the artery is slightly under higher pressure. Okay, these very low pressure. But the point is the liver is equipped with thousands and thousands of capillaries, and those capillaries are relatively large. When their capillaries are large, you call them as sinusoids. So, there is hardly any resistance. So, the blood can easily flow into the liver and through the liver into the central vein, and from the central vein into the. Now, I just want you to be introduced to one very interesting cell and a name, whose name I want you to know that name. So what do we have? We have a wall of liver cell. Wall is a bad word, but I will still use it. Another wall of liver cell. Are you with me? And in the middle you have what? A capillary. In the capillary you have a very special type of cell. I don't know. Yeah, they have. This is called as a Kupffer cell. Kupffer. He was a German scientist. He discovered those Kupffer cells. Say that again. Again. Kupffer. Kupffer cells are phagocytotic cells, and obviously, you know, the material is coming from the intestine, along with what? Digested food. Along with the digested food, what all viruses and bacteria and all that can come. Are you with me? Their microbes are also very smart. Okay. So, the Kupffer cells is within our body. It is one of the lines of defense. Okay. Are you getting my argument? So, if anything foreign that enters which should not have entered apart from food, again it will be consumed or it will be dealt with by the Kupffer cells. So, remember the name Kupffer cells and appreciate their importance, Kupffer cells. Okay. Now, this is whenever either for two reasons. Look at the two healthy liver, okay, and look at that liver. I put an extreme case there. That is called as a liver of a person who is suffering from liver cirrhosis. Now, liver cirrhosis can happen under two conditions. One is regular consumption of alcohol for a very long time. Okay, it results into what you call as liver cirrhosis, or some infection. Okay, some hepatitis or some jaundice or some, both of them can cause. Now, what happens when you get one of the liver cirrhosis actually happens because the infected liver or the regenerating liver gives rise to a lot of fibrous tissue. You remember we talked about elephantiasis. Why does the leg of a patient suffering from what? Okay, yeah. Say that again. Perfect. Because of that. So, that similar fibrosis happens here, and that beautiful anatomy of your liver you have in the liver, okay, is messed up. Are you with me? And then the efficiency of liver goes down to 10%. Everything may be, all the cells may be there, they may still be manufacturing bile, but the anatomy and the efficiency, you see, every day within a period of 24 hours, our liver secretes almost 300 to 400 ml of bile, which is a lot. What did I say? Our liver secretes how much of bile? 300 to 400 ml of bile, which is a lot. That efficiency goes down in what you call as the cirrhotic liver, and okay, good. Let's move on. So, what does the bile do? It helps us to, okay. Now, do you all understand the meaning of the word, what word? Emulsification of fats. What is emulsification of fats? So, I take a beaker, in the beaker I take half the beaker of water, on the top I put some oil, okay, and I find that the oil makes a layer at the top because its relative density is less, lighter than water represents, and I stir, nothing happens. I mix, then again they separate into layer. Now, I want to make sure that I want to somehow mix them, they can't dissolve, out of question. Then what do? I put 1 spoon of detergent there, a soap, okay, and then stir it. Okay, and as a result of that, then I find that the fat now is now broken into tiny pieces. Okay, and then it can uniformly spread in water. And now what I say is I have an emulsion of oil in water. What do I have? Emulsion of oil in water, and the best example of emulsion in water is the milk. Okay, is the milk. So, when I make curd out of that milk, I am breaking the emulsion. Are you with me? Hello, that is an emulsion. Okay, blood is an emulsion. Okay, see the particles of different densities are spread around. Okay, because of the detergent action. Now, one of the very important function of the bile is to okay, just okay. This is just to, we are talking about the gallbladder. I'll come back to that. Come back to detergent action in a while, but let me address this slide. We are talking of a gallbladder. The bile comes into the gallbladder. The gallbladder does a lot of processing. Gallbladder absorbs a lot of water. Okay, it's good that concentrates the bile, but if it over concentrates, okay, then the materials crystallize out, and once they crystallize, then they can give rise to stones. If the stone is there, then it serves as nucleus for formation of, for making the stone bigger. Okay, but that's the thing we have to leave. And then this is the can you see there's a dotted line, and then can you see the red arrows? What do they represent? They represent the action of cholecystokinin on the smooth muscles of the gallbladder. Okay, so it constricts and then it actually pumps the bile into the bile duct, and then from the bile, and this will happen under the signal that it gets from the CCK, and the CCK will be released when fatty food, okay, or amino acids, they arrive from the stomach into the duodenum. Okay, the cells will release the CCK, and so CCK is a key factor that will determine the timing at which you want the bile to arrive in the duodenum. Good. This is just to show you the structure. It's a peptide. What peptide are we talking about? Okay, this is just to show you this is the bile as it is being secreted by the liver. Then the bile may either go directly to the duodenum, okay, as it happens when you are actually having food, or when you are not having food, the bile secretion may go on, and then the bile may be stored in the gallbladder. While it is stored in the gallbladder, it undergoes a lot of processing. For that, I'll just draw your attention to the bile salts. Bile salts. How much is there in the freshly secreted bile? How much is it? And then how much is it in the gallbladder? So you see, lot of processing is happening. Okay. Another function of the blood is hemoglobin is being broken down. Hemoglobin being broken down at the level of spleen. Okay, and the broken molecules they give rise to what you call as bilirubin, and bilirubin is got rid of out of your, to throw it off your body. It goes into the bile, from bile into the undigested food, and by way of feces it is thrown out. And then let me move on. Okay. Now I am going back to the earlier step. What was my earlier step? My earlier step was, how does bile serve as an emulsifying agent? Why do you need that? Because you see, if you eat a spoonful of butter, okay, and if that spoonful of butter, more or less like spoonful of butter till it goes to the intestine, it will be very difficult for the enzymes to act on it. So to make sure that your enzymes, which are say lipases, which are secreted at the level of duodenum, they have access to every tiny bit molecule of fat. So the least you can do is to break down fats into tiny bits. Are you okay so far? And for that we need a detergent, and detergent comes from what you call as bile salts. Bile salts. What did I say just now? Understand, bile salts. Now let's see how the bile salts are formed. Okay. Now first thing I want you to remember is this structure, this molecule called as cholesterol. It very widely distributed, it is a part of the plasma membrane everywhere. Cholesterol, it is everywhere. What is cholesterol? It is a molecule, it is a steroid. What is it? Cholesterol, it is a steroid, okay. It is made up of 4 rings, 6-membered, 5-membered. Got it? Call them as what? A, B, C, D. Look at the arrangement, A, B, C, D, and there is a side chain. And the entire molecule put together has 27 carbon atoms. How many? Remember this, okay? You can't be a student of physiology. Your cholesterol has to be remembered, okay? When we eventually talk about, which I will hopefully in another week or so, when I talk about the steroidal hormones, okay, like the hormones of the adrenal cortex and the sex steroid hormones, it's all the story. All the stories begin with cholesterol. Okay, so this cholesterol. Okay, from this cholesterol, this side chain, okay, it is acted upon by an enzyme, side chain cleavage enzyme, whatever, and then you get 2 compounds, which one is called as cholic acid. The similar, but only some part is cut away from here. Are you with me? And so what are you getting from cholesterol? Cholic. We are capable of synthesizing cholic acid. I am sorry, we are capable of synthesizing our own cholesterol, but lot of it comes from our food. So both the sources are there. Okay. So you have it is converted either into cholic acid, or this is chenodeoxycholic acid. Are you okay so far? I am talking of two molecules. Okay. Okay. What are the two molecules I am talking about? Cholic acid and quinolactone. They are both derived from where? Cholesterol. Cholesterol. Okay. And where is all this happening? This is all happening in the liver. We are on the hepatocytes are doing it. Okay. Now what happens is these two are fats. Cholesterol. Cholesterol. They are lipophilic molecules. Okay. They can go anywhere to anywhere and they have no charge. Are you okay so far? Now, one interesting thing that happens is these two molecules combine with amino acids. And what amino acids are generally chosen for this purpose? The two amino acids are largely glycine and to some extent taurine. Are you with me? And these amino acids, these two amino acids then combine either with cholic acid or with this what is it, quinodeoxy, whatever it is, and then you get what you call as, what is it, taurocholic acid, and what is it, whatever it is, you remember the name, I don't care. Okay, you got it? So that amino acid now, that amino acid has been hooked in here and that amino acid has been hooked here. The moment that happens, you call them as bile salt. What do you call them as? Again, what do you call it as? Bile salt. Now here is the beauty. The beauty is because of the amino acid, can you see NH here? Hello? NH here, so nitrogen is introduced for, because here and because of here there is a charge. So you have been able to have a molecule in which one end there is a charge, at another end there is no charge. That is the beauty of biology. Hello? Charge at one end and no charge at another end. So that no charge at another end can have freely conversation with the fat molecule, and the other one can have free conversation with the water molecule. Are you getting the story now? So these molecules now, so what am I talking about? I'll just use a word common, bile salt. What will I use the word? Bile salt. By that I mean what? This compound and these two compounds. You know how these compounds have come into being. They are all derived from cholesterol. Done so far? Now, yeah. It's amino acid. It's not among the 20 amino acids. No. It's not among, you are right. It's not among the 20. Like GABA. It's not you can have n number of, when we talk of amino acids, 20 are the basic blocks for protein. They are beyond that. Now, here is a clear story. The author shows the molecule being divided into two components for our understanding. That yellow part is the earlier part derived from cholesterol, and there is no charge on this, and this is that glycine has been added here, and this red one is what? Okay. So the red one is called as what? Okay, because there is charge, it is what? And this is what? Okay, now if you remember this story, okay, we are again talking about is this picture very clear to you? This water at the bottom, oil at the top, you have added detergent, as a result of that you have finally what? An emulsion. What emulsion is there? Emulsion of oil into water. Very good, and that is because the detergents give certain charge, okay, on those oil droplets, and then it can form the emulsion, and as a result of that we are capable of having this organization in which we have the lipid molecule, not lipid molecule, tiny droplets of lipid, not a single molecule, tiny droplets of lipids, and they are being surrounded by the bile salts. So each one outside there is a bile salt, and it is organized like this because this is the hydrophobic side which comes in contact with the lipid part, and this is what part? Hydrophilic is on this side, hydrophobic is on the side of the lipid, and then it forms a cover, so that now this although it is oil, okay, it can move freely, it's very small, it is now once, and this is also what you have now. What will you call once this happens in the intestine, what will you call it as? It's emulsified fat, okay, and each such fat droplet is called as a micelle or micelle, okay, MICELLE. It's tiny. I want somebody to read the line in the blue font, please. Can somebody help me out?
Ordinarily, the micelle is in the duodenum have a diameter around 14 to 30 microns.
Very tiny. Can be, you can't even see it with an eye. Okay. Now Now, duodenum is also secreting lipases. Okay, so there is a micelle there. Can you see there? This one, and these are the bile salts. Okay, and here is a lipase entering into the micelle. Okay, so the lipase will be released. It will act on this fat. What is this fat? Triglyceride. Okay, and then this triglyceride will be broken down into what? Broken down into monoglyceride. Okay, and fatty acids. Okay, I forgot fatty acids, monoglycerides, cholesterol. It will be broken down, and then you will get the, and then this can be absorbed across. Okay, so yeah.
Are the bile salts also helping in the formation of smaller fat droplets?
Actually, yeah, it is. Actually, it is the agitation at the level of this. Lot of that I didn't talk about it. The as a result of, we know peristalsis. Okay, at the level of intestine, the aim of the peristalsis is not only to move the bolus of food along, but also to bring about the agitation. Just move. And how it does is, this I will put a tube. Okay, and then I will press it, release it, press it, release it, press it, release it alternately. Not that I am moving the food forward, no. Okay, I will do it after 5 minutes. Are you getting my argument? Hello, press it. So, the food goes here, press it, so it may go back actually. Okay. What I am really doing is facilitating the free movement of the that enzyme we saw earlier one, or all the enzymes. Okay, particularly so that they have so that actually point number 1 is break down the big chunk of lipid into tiny micelles which are so small. How small we have seen in the previous one. Okay, and then in the process bile salts are there, so the bile salts will form there, and then so we are actually digesting, we are taking, and then it takes time, maybe 6 hours, 7 hours, 8 hours. It is a slow process. Okay, and during that time the fats are being digested, so that they, okay. So here we have a part of the intestine from the stomach. These are the large circles are what? Fat droplets. In what form are they? Triglycerides are there from the bile duct. Okay, you are having what you call as the bile salts. The bile salts combine, the lipase is put in there, is introduced there, and then you get the micelles which will play an important role in the. Okay, listen to this. Liver has spent lot of its energy in synthesis of bile salts. Okay. And the nature is economical. Okay. So, what trick the nature is playing here is, once a bile salt is released in the duodenum by way of bile, and then it has played its function in the intestine. What function? Emulsify the fats. Okay, emulsify the fats. Then the nature has said, can I recover some of those bile salts? Get the problem? If I can recover those bile salts, I do not have to go through the trouble of making the bile salts once again. Are you getting the argument? Therefore, the nature has come up with a very interesting phenomena which is written at the top. Please read for me. Enterohepatic circulation of bile salts. So, let us see what is the story. So, this is a part of the intestine, the fats have come, the bile salts have come via the bile duct, and so here somewhere in the intestine what all we have seen in the earlier slides has happened, and the micelles have been formed and they are ready to be absorbed. After that function is done, there in the wall of the intestine, in the latter part of the intestine, we have a pump. What pump it is? Secondary active sodium symport system. Okay, so in those cells there will be ATPase somewhere on the basal side, on the apical side there will be a symport system. That symport system now it specifically picks up the bile salts. Okay, it puts them in the hepatic portal system, and the hepatic portal system will carry the molecule bile salts back to the liver, and those bile salts will be reused. Are you with me? What is it called as? Enterohepatic circulation. Look, I am going to tell you something very funny. I have been teaching this very clearly to the best of my ability for last, I do not know how many, I will not tell you how many years. And In the examination, I ask a question, describe in brief the significance of the enterohepatic system, and half of you will write hepatic portal system. Hello, are you awake? It is so irritating for me, you know. No, not that you are awake is irritating for me. No, I am good with that. Okay, what's my problem? You see, I shout hoarse my word, trying to explain to you the difference between what and what? Hepatic portal system and the
Hepatic circulation.
they are two different things. They are two different things. But when I ask a question, this. Generally, hepatic portal system is fine. But enterohepatic means people will write. Half of you, even now I challenge you. Half of you, I'll tell you, I'll ask a question. Maybe I do, I don't. Half of you will still write. What is described the enterohepatic system? Hepatic portal system. Think, okay. Have I given you enough dressing down so that you don't make the mistake? Look, all your seniors have done it. All your seniors. Okay, good. So I think I will not teach you the rest of the things where I talk about the absorption of glucose. Very simple, we have done kidney, so you can do this here. Absorption of fat, how the micelles and the fats are absorbed, and how the amino acids are absorbed, and with that I will come to the conclusion of our topic on digestive system, and without wasting even 5 seconds, I will move on to the next topic.